Hello, I'm Colleen Yanarella. I'm a bioinformatic and computational biology PhD candidate at Iowa State, and I work on spoken descriptions of phenotypes in the field. Plant breeders have been working to produce plants that are more robust and higher yielding in different environments. These are pictures that I've taken over the past year of different fields in about a 2-mile area. Last year in our field experienced heavy hail in pop-up storms that were about the size of a quarter. The same field experienced drought where the ground started cracking. And there were different pressures like weeds and insect pests that were impacting the plants. And more recently, there's been some extreme flooding in the area where even the plants on the edge of where it flooded began to turn yellow. Last summer, this was a very high moment for me where we had participants and volunteers out in the field taking smoke-in recordings of plants in the field. And I'm more used to an environment like this where I'm in an air-conditioned room working in front of a computer. But last year I had a mobile unit where I was out in the field making sure that all of the recordings were not corrupt. And I'm going to discuss the methods that we've been using in the field and how we're using this data and applying it. To begin, let's discuss how we perform association studies. Then I'll discuss how we collected these spoken phenotypes, an overview of our pipeline, and the next steps. The first thing we need is founder lines. And after many generations of intercrossing, we can get what's called a natural diverse accessions or association mapping panel. And these have diverse genotypes. We plant these in the field and we do a process called phenotyping for our traits of interest. And over on the right, we have 3 rows of green plants and 3 rows of brown plants, which align to each of these accessions in our mapping panel. And we get genotypic data where we genotype these plants. And this is becoming less and less of an arduous process, and phenotyping is becoming more and more of an arduous process in comparison to genotyping. And in this instance, we have 5 what are called SNP IDs. So these are single nucleotide polymorphisms. And if we look at our row of phenotypes or a column of phenotypes, we see that there's 3 green and 3 orange. And if we look at our SNP ID, we might think that there's something like a dominant recessive relationship happening here. But when we actually do this in practice, we get something more like this, where the top 3 rows are all green plants, and the bottom 3 rows are a mixture of brown and green plants. And the goal of performing association studies is to determine marker trait associations. And in this case, our markers are our genotypes or our SNPs, and our trait is the plant color trait, and the phenotype is the color. The diversity panel that we used was the Wisconsin Diversity Panel, and this is a drone shot of above our field from last year. We planted the Wisconsin Diversity Panel in 2 reps, Rep 1 and Rep 2, and these 2 reps were randomized independently of each other. There were 686 Wisconsin diversity lines, 8 B73 environmental controls, and 25 positive controls in each of the reps. The 25 positive controls were seeds ordered from the stock center that, if grown in the right environment, would have very interesting phenotypes. And these phenotypes were really deep purple plants or feminized tassels or bushy silks or things like that that would be very obvious if they were in the correct environment. And we did this because we were having people go through and describe the fields in their own words. And these were things that people would be able to describe if they were present and visible. And these reps were composed of rows. Each row was an accession or a genotype from one of these 3 categories. We had 720 rows per rep. And if you add these up, it doesn't equal 720. After we packed the seed and planted it, we realized that we planted one of the Wisconsin diversity lines twice in each rep. Each rep also was built up of ranges, which were 60 rows. And this was important for how we collected our spoken phenotypes, which we're going to discuss next. But each range consisting of 60 rows, we broke these up into 3 recordings of 20 rows per each recording. And we did this for just in case somebody forgot to hit record, or if the data cut out, or if a microphone came undone. That way there was less that the students would have to repeat. How do we collect spoken phenotypes? Well, in addition to collecting spoken phenotypes, we collected quantitative measures and scores of traits of interest. And we did this by going through with pen and paper and measuring sticks and measuring and scoring different plant parts. And we also went through and collected spoken recordings using small Sony recording devices and lavalier mics, or if students didn't want to use lavalier mics, they just held a recorder up. These students we called Delta, Golf, Kilo, Lima, Mike, Quebec, Victor, Yankee, Zulu. We also had additional volunteers go through and make recordings, and we called them Alpha, India, Tango, and Whiskey. I could tell you that I was Whiskey, but I can't tell you who any of the student recorders were because we have an IRB. It's an exempt status IRB. And we agreed to hold ourselves up to the HIPAA standards for data storage and use, which means that we cannot provide the raw recordings publicly. And this is to protect our student participants. And the other reason we use these NATO code names is because it allows us to determine the row number. Without the use of QR codes or anything like that, we can just parse using the keyword of the NATO code name and then determine the row tag number for the recording. And this process still allows us to be fair, findable, accessible, interoperable, and reusable. While we cannot release the data from the raw data recordings from our student participants, we can for the additional volunteers. And the student participants went through the field in 3 passes. They went through the field 3 times in its entirety. And the additional volunteers went through the field for once. They made 1 pass in the field. So we still have a lot of data that we can make publicly available as we finish up our pipeline. Next, let's discuss how these recordings worked and the equipment that the students went through in the field. In addition to their recorders, they were given small cards that were just folded paper that included a reminder to hit record and to say their NATO code name and row tag number at the start of each observation. These cards also had diagrams of corn plants and required traits that we wanted them to talk about if it was present or visible, and additional observations that if present and visible to describe. We did this because a lot of the students had never been field researchers, especially with corn. So this allowed them to go through and talk about and describe certain traits without forgetting what the terms were called of what they were describing. We gave them more detailed diagrams of the ear, tassel, and leaf blade, but we didn't try and manipulate the way that they were describing about the plants as far as making the descriptions. We wanted them to use their own words to describe the plants. And this was because we didn't want them to act like computers or robots. We wanted them to act like human beings describing plants. And we didn't want to limit the vocabulary that they were using as far as descriptions. One way we did manipulate them is after their first pass of the field, a group of students was asked to make a general description of the plants as a final statement for each row, and another group was asked to make a general description of the plants in a row right after the row tag number. And we flipped them for the last pass, and the students who started with describing the plants overall as a final statement disliked being asked to say it directly after the row tag number because they preferred to make tiny descriptions and then make a judgment about what the plants look like overall, rather than just make a quicker judgment about what the plants look like overall. So that was an interesting thing to hear from the students. The next slide is about a 1 minute long video of what a recording looked like in the field. And I'll pick up after the recording's over.
Whiskey 1574. These plants are extremely tall. They are lodged. The leaves feel veiny and a little rough. There's pink around the edge of the leaf blade and down the margin of some of the leaf blades. There's also pink patches up the plants. And there's striped purple brace roots. The ears are very large. And the tassels have a few branches, and they droop over a little bit, especially at the ends of the branches.
That's an example of a recording for one row, and I would continue down the range until I hit 20 rows, and then I would have stopped my recording and then continued on. One thing I'd like to point out is, if you notice, I was talking about the feel of the leaf, and something that's interesting about this sort of research is describing the feel of a leaf. Juvenile leaves feel softer than older leaves. Older leaves feel more like sandpaper. And that's information that we didn't express to our students, but they started describing the feel of the leaf and started picking up on different things like that. And that relates to how we generated our pipeline. The top portion is the process, and the bottom portion is an example. The left in the purple boxes, we have our audio processing component, and on the right, we have the association study component. We record speech for plants of each genotype in the field. Whiskey 1574 follows the example of the video that I just played. And we're using AWS, Amazon Web Services, Transcribe. And this allows for a tuning file, which is a text file for tuning for scientific and agricultural terminology. And this is really effective for homophones, which are words that sound the same but have different meaning. An example that we ran into was rose versus rose, where AWS was selecting rose as an ROSE, the plant. And we wanted it to select ROWS, rows of plants. And the tuning file allows us to do that. And we work with the IRB office to generate a protocol which allows us to push our data to AWS, process it, transcribe it, and immediately delete it from their servers. And we have scripts that do this. And we get text, which we can compute on, which leads to the association study component, where we are generating phenotype networks based on similarity, and this is semantic similarity or word meaning similarity. And we're performing clustering and thresholding to identify what we call synthetic phenotypes. And I will provide an example of a hypothetical synthetic phenotype network on the next slide. And we're going to use these synthetic phenotypes to perform association studies. An example of a hypothetical synthetic phenotype network would be something like this. On the top, we have short internodes, compact plant, short plant, broad leaves. On the left, we have bushy tassels, large tassel glooms, clump tassels. On the right, we have smut present, and then we have colored alurone, purple brace roots, and anthocyanin on leaves on the bottom. And there's X in the center node, which indicates that this network is much bigger than this. And we'll perform thresholding to determine what a synthetic phenotype is. So synthetic phenotype 1 is short and internodes, broad leaves, short leaves, compact plant. And these nodes all mean short plant. And synthetic phenotype 2 would be colored alurone, purple brace roots, and anthocyanin on leaves. And these have something to do with purple or pigmented plants. And bushy tassels, large tassel glooms, and clump tassels would not have been above the threshold to be considered a synthetic phenotype. We'll pair these synthetic phenotypes with the genomic information that we have available so these are SNPs for the Wisconsin Diversity Panel, and I'll discuss this more in the next steps, to perform association studies. A set of metadata that we have relating to these descriptions in the nodes are the rows which these descriptions came from. And we know the genotypes for each of these row numbers. Next steps. In 2019, a paper on the Wisconsin Diversity Panel was published, and they used 942 Wisconsin diversity lines, and they generated a data set of roughly 900,000 SNPs. They were looking at biomass traits, and one of the traits was plant height, and they identified 4 SNPs of interest. Our collaborators at Purdue, Brian Dilks' group, he has 2 postdocs, Raj and Aman, who've worked extensively with this SNP data set. And they've been using a tool called HapMap3. And what HapMap does is it imputes missing SNPs. So it uses statistics to determine SNPs that are missing in the data set. And using HapMap3, they generated a data set with roughly 55 million imputed SNPs for the Wisconsin Diversity Panel lines. And then they performed filtering, and they kept all of the SNPs with a minor allele frequency of greater than 5%, which resulted in a data set that I have access to that has roughly 22 million imputed SNPs. And these 2 data sets I'm going to use to perform association studies using our numeric data and our speech-derived data. Therefore, I'm taking spoken descriptions from the field, transcribing them to text, and performing association studies. I'd like to thank all of my lab members, Brian's group, my funding, everyone who helped out in the 2021 field, and our student workers and additional volunteers. And I would like to thank the Digital Ag Special Session for inviting me to present this talk. Thank you.